Hi, everyone. I'm Heather Gatney, and I'm the coordinator of the CVFS webinar series. Our host today is Coulter Mitchell, Research Associate Professor at the Survey Research Center at the University of Michigan. Today, Coulter is going to tell us about polygenic scores in the CVFS. There will be a Q&A session after the presentation. We ask that you remain muted until then. During the Q&A, you may raise your hand to ask a question or use the chat. Also, please feel free to use the chat during the presentation if you have a clarifying question. We'd also like you to know that this webinar is being recorded and will be available to view from the CVFS website. We'll also put the slides up there later. Okay, Coulter.
All right, I'll take it away. So I'm gonna go ahead and start sharing. So everyone can see that, right? So there's a couple things. One, I know we typically do like a full lecture and then we have kind of interaction at the end. That's still going to happen, although please jump in at any point if you want to talk more about something or have a clarifying question, as Heather mentioned. But actually, the last section, I was hoping we would have enough time to maybe go through um, some scenarios at which we might be able to use this, if that makes sense. So I think one of the things to also know is that I've already given one presentation on kind of polygenic scores and why they're interesting and why how they're constructed and things like that. That's on the website. So this time it's more about application of polygenic scores. And part of that will be a reminder of some of the basics. I'm thinking about life course processes and how that might relate, environment and gene environment interactions, a little bit on instrumental variables. And then, as I mentioned, I really want to talk more about the Chitwan Valley Family Study and kind of maybe thinking through. And I'd be happy to do this in a lot longer detail with anyone who's coming up. Oh yeah, thank you, Emily. Um, yeah, we'll talk through some of these other potentials. All right. So the other thing to know, and I think I saw Aaron's name pop up here as well. So they're actually, if you really end up becoming more interested in this, whether you're watching now or later on online, there is a course that we teach here at the University of Michigan called Genomics for Social Scientists. And it's an introduction. It's a whole week here at the University of Michigan in person, this year at least, June 10th through the 14th. And we have travel stipends, and the applications are due February 19th. But again, if this is something that you think you might actually want to learn a lot more about in detail, please come. It's a great opportunity to meet lots of other people and talk about your ideas. Okay, so just to tie back to kind of that first lecture, to talk just a little bit about why people even are interested in genes. Um, some of the key things are genes don't change. So imagine a predictor that starts at birth is the same, and yet it actually is predictive of many different outcomes. Um, and you can measure it once, and you don't have to try about getting it multiple times. That's a very novel part about genetics. It's actually relatively inexpensive to get at this point. And as we'll talk about today, is pretty useful in several different applications. The other thing, and this is going to tie back to, again, that first presentation, we actually have a lot of exploratory data analyses of genetic data called genome-wide association studies, or GWAS, if you've probably heard that before. Large consortia, several hundreds of authors sometimes, who all join together and do these exploratory data analyses of genetic variants, sometimes millions of them, and to see which ones are the most associated with different outcomes. And just about any outcome you can imagine has probably had a GWAS on it. Um, Anything from educational attainment to type 2 diabetes to common diseases, even divorce and things like that. So just about everything you can imagine. So when we create a polygenic score, we're taking the results from those external studies and applying them to our data. So, for example, this is called a Manhattan plot. This is the chromosome. The x-axis is the chromosome location, the address of that genetic marker. And then you have the negative log 10 p-value. So the higher it is, the smaller the p-value. And that's the y-axis. So you can imagine if you take something that has a really high significance, so very small p-value, they're often larger effect sizes. If we take each one of those as a weight, we multiply it by their gene, we add all those together, we get a summary score for each person of kind of how how strong, well, not how strong, how predictive their uh, gene might be for a certain disease, right? So they might be higher, they might be lower. It's not necessarily causal. We'll get to that later, but it is, in other populations at least, associated with it. And that's one of the things that we talked a lot more about in the first. But just as a reminder, this is essentially a predictive algorithm um, of some other outcome or uh, environment even. So that's all it is, just a predictive score. And typically what happens is as you get more and more of these, if you just had one or two genetic variants, you could actually have a pretty skewed distribution. But as you get more and more, thousands or millions, it becomes a fairly normal distribution. And that's what you end up seeing, is a fairly normally distributed predictive variable that doesn't change over someone's life, um, and can sometimes be very predictive of different outcomes. So, as I kind of mentioned, again, this is mostly still the first lecture. There have been GWASs on many, many traits, and I can't stress that enough. So many traits. There's whole databases on all of the GWASs that have been created. Um, and as we've gotten bigger studies and maybe better measured studies, we've gotten larger and larger effects and therefore better prediction. Um, Although it's taken a long time, we're actually starting to get a little bit better about being able to predict within different ancestries originally, and still for the most part, it predicts mostly in European ancestries. That's where the GWASs were conducted, but that's slowly improving. And one of the things that is really great about Chitwan Valley Family Study is that it is a very unique study compared to all the other GWASs and genetic studies that are out there. So it's kind of a unique opportunity to take things that were created in other populations and see if they predict in this one. And finally, there's still a lot of work on trying to improve the interpretation and understanding of kind of what these polygenic scores are. And that's actually a big part of what we'll talk about today. Okay. The other thing to kind of know is that this is always the case whenever you start thinking about genetics, um, that you typically have this interesting thing of the fewer number of genetic measures you have, the better your understanding of biology. And I think that makes sense. If I were to say, here's one measure, one genetic measure, and that's it, one gene that I've measured, I probably could understand a lot about the biology of that by looking at animal studies and other things like that. The instant that I start combining across the entire genome, I start losing my ability to give any biological interpretation. So, and that is where most polygenic scores typically lie. Typically, they're not just taking one or two genetic measures, but hundreds, thousands, or even millions and combining them all. So they might be more predictive, but you're going to lose some of the biological insights. So that's often the trade-off. And for most social scientists, they're willing to do that because they're not as interested in the actual underlying biology. If you go talk to a molecular biologist, they're going to want to use uh, more gene specific, more biological data. So that's essentially what this is saying. Um, uh, yeah, and again, it's fixed across life course, and there's no way to have reverse causality. So that's an important thing to keep in mind. It's rare to have predictors that are like that. So what I'm going to talk about today is three kind of promising avenues for incorporation of polygenic scores. And so in some ways, I almost want you to think about the study that you typically do and work that you do, and maybe then we can talk through anything specific that you have. But first is life course. So you take a polygenic score that was probably developed in adults, and you see what are the processes that maybe lead someone to that. For example, if you have a polygenic score for type 2 diabetes, can you see the effect of that even in childhood? Right? Because remember, genes don't change over time. So do you see it kind of predicting a certain progression or different other activities even when they're kids? How are genetic liabilities stratified across different environmental exposures? That's a key thing that a lot of us, I think, are interested in as social scientists is, do we see selection into different environments? Do we see interaction between different environments and kind of underlying genetic predispositions? And the last one that I won't spend very much time on because it can get complicated really fast. Um, there is a random component to given your two parents, which version of the gene that you get um, is random to some degree. And so between you and say a sibling, which sibling got which set of genes is fairly random, and that same idea can be used to try to create an instrumental variable for different phenotypes to kind of sort out causality of different uh, social environments, but also different phenotypes. Okay, so let's go back to kind of this um, imagining like how can you take a polygenic score that's been created in some other population and examine what it might look like, its effect on different processes. Some might say developmental processes, life course processes, depends what field you're on, field you're in. So one example of within the social sciences that has received a lot of attention is an educational attainment GWAS that then was used to create an educational attainment polygenic score. So it's exactly what you might imagine. How far did someone go in school? Did they complete college? Something like that. Can't really study that in kids because they haven't completed school yet. So this is really, these scores were originally mostly done in European ancestry populations. And it was people who've completed, are old enough to have completed school or not, right? So you might ask, why would that ever apply to kids? How could it apply to kids? Well, in fact, when you take that score, because we can take those weights, as I said before, and create a score in another study, in this case, a study of children, you can then start to measure, are there differences at from birth in different polygenic scores, high or low in education, um, and things like smiling, sitting up, walking. And in fact, what you do see is that those who have higher polygenic scores for education seem to talk faster and also communicate in sentences. But pretty much everything else are the same. So you start testing these things out. Now I'm using education, but you can start thinking through these other ones. Again, type 2 diabetes, what would that look like? Are there any earlier processes that might happen? Or you can look at something like later life and even control for how much education you had. Does the genetic predisposition to higher education have any effects? And it does on in terms of pension and wealth and even longevity. So how long you live, even controlling for your actual education. So again, these are thinking through how you might take a score and kind of find out the intermediate steps that might lead you to an outcome that you're interested in, or a parallel process for something you're interested in. You might also just be curious, how big are these effects? Most of the time, I'll be honest, they're pretty small. They're very small, in fact. For most polygenic scores, they're very small, and they tend to be even smaller if they weren't created in the population that you are testing them in. So in CVFS, most of them are going to be fairly small, like less than 1% of variance, less than half of a percent even. But as things are getting bigger, models are becoming better, phenotypes are becoming better measured, that's changing. So as an example for education, um, in fact, if you look over here on the model on the right, you see it's all kind of shaded except for the one that's the education polygenic score. So it actually predicts more educational attainment um, than household income, but a little bit less than verbal ability. So that starts to give you a sense of how big they can be. For some traits, they're quite high, like height is quite high. Um, and this is actually pretty high already. Most other complex diseases and social variables are nowhere near this, but that gives you a sense of kind of what the upper bound is. Um, yeah, we can talk more about this later on if people are interested. Another thing you might ask is how robust is the prediction? So this is from another paper that just was looking at how, again, I'm sticking with this theme of the education polygenic score because it's one that's talked about a lot in the social sciences, is just seeing how that score predicts even within 4 different studies, and within those different studies, different strata of socioeconomic status, right? So you could imagine a situation where it actually is not at all predictive among some groups or in some studies, but very predictive in others. So the 4 different studies were the Ad Health, Dunedin, which is a New Zealand cohort. If you haven't done it, it's a birth cohort in New Zealand, Wisconsin longitudinal study, and the health and retirement study. Um, and the x-axis in this case is that education polygenic score. Higher score would mean higher educational completion on average. And then the y-axis is the socioeconomic index, or wealth, I think is what it is for HRS. So how do those compare? And you start to see that, in fact, it's actually fairly robust, fairly strong across all of them. A little bit different with Ad Health, lower SES, but for the most part, very robust. Now, that doesn't mean that all polygenic scores will act that way, but this is an example of kind of what you could be thinking through. Like, is prediction the same in, for example, CVFS in each ethnicity, right? Each caste, or is it different in different regions? Is it the same in city versus rural? You could imagine several different versions of this that you would want to make sure it's robust to see if there's maybe some other confounding going on. One of the things that I think is particularly noteworthy for CVFS is, and we'll talk again more at specifics in a little bit, is there's a family design here, and because of that, you can actually take polygenic scores of siblings, which, as I had said before, there's a random component here of which siblings got which genes, and what's the effect on an outcome. In this case, it's comparing siblings in education, occupation, and wealth in different cohorts. E-risk is a twin study. Ad Health has twins in it. WLS has siblings in it. And you start to see there are actually pretty sizable differences for some of them in terms of if a sibling got slightly higher polygenic score for educational attainment, they actually went further in school on average. So there is a causal component to this that you can start to tease out using sibling models and family-based models. And again, imagine now you have several different family-based models in CVFS. You could start to examine some of these things too. Is there a causal effect of a polygenic score? Okay, so that's kind of teasing out what I was saying before, these life course processes, the application of a polygenic score and kind of pulling it through all these different steps that might lead to a certain phenotype. A slightly different role is examining how it is correlated with or interacts with the environment. So it's probably not surprising to any of you that if you were to conduct a discovery analysis, like a GWAS, as we were saying before, and it's that outcome variable, like education, is actually socially driven or socially contextualized, that it will probably, when you start doing these analyses, it won't just pick up biological underpinnings of education, but it'll pick up some of the social correlates as well. And that is absolutely true. There is a baked in kind of social component to a lot of these polygenic scores. So it's kind of critical for two reasons. One, please don't assume that this is the biological truth, because it is not. None of these are biological truths, right? Um, there's a mix of biology and social stuff being mixed in. The other thing to keep in mind is now you have a pretty good um, set of analyses to run and seeing what environments they are correlated with. How can we maybe tease out some of that biology from the social science? How do we pull that apart? Or is it possible that some of these polygenic scores are all social? And in fact, there's very little biology mixed in. And there are people who are trying to tease that out as we talk through these. So let me show you a few examples of this. Um, so here's one that kind of ties into this a little bit. It's called genetic nurture or genetic nurturance. I find this one fascinating. I pulled this one up in part because, again, something that can be done in CVFS that can be done in a few other studies. So that is that you can imagine that uh, a, a parent passes on genes to their kid, and their kid then has some sort of outcome. In this case, if we use educational attainment again, um, a mother or a father might have a certain score, a polygenic score for education. They pass half of that down to their child. The other parent passes the other half down, and that creates their own profile, which is then predictive of their outcome. The interesting thing is that we find that parents, even controlling for that kind of indirect effect, parents still have a direct effect of their genome on the child's outcomes. And why would that be? That's in part because um, those untransmitted genetic measures are still having an influence on which environments people are selecting into or creating. So in this case, Jasmine Wirtz wrote this great article that then shows that parents or mothers' polygenic scores were predictive of several different parenting outcomes that explained why the parents' genomes were still having an effect on the child's educational attainment. That makes sense. So there's still kind of a parenting component to this that's still connected to genes. So it's a genetic nurturance is a different way of talking about it. But you could also imagine selection into different environments would be the same. So this is more of an active, we talked about that last time, an active gene environment correlation, where they're creating it, but they could also be placed into different environments, evocative. And this kind of ties into kind of a broader picture of environment, where you could do a few different things. One is thinking carefully about the environment in which you're studying, and where genes might or may not apply. So we have a couple different versions of things here. One on the left is actually looking at different uh, genetic score and how that interacted with actually um, whether or not you were drafted for Vietnam. So Vietnam conscription, which was in theory somewhat random, at least the date at which, whether or not you actually went to Vietnam was a different story, but whether or not you were selected for in the draft was random, and genetic potential for educational outcomes, and then how far you went in school. A slightly different version of that, that's more trying to get at a gene environment interaction where the environment is random. A different one is maybe thinking about the way education. I'm sticking again with the educational polygenic score, but it can apply to anything. Um, how how strong that effect is in pre-Soviet era or in the Soviet era versus post-Soviet era, and allowing people to go to school or not go to school, right? So a big change happens, and suddenly the genetic uh, prediction goes up. You actually see this. There's another great paper that was in Estonia. There's another paper done on, I believe it's WLS. Pam Hurd wrote it, that showed the effect of just time in the United States on women's polygenic score and how far they went in school, which essentially showed that as society became more um, tolerant of women going to school, for good reason, obviously, uh, that genetic prediction of going to school became much stronger for women. That makes sense. Uh, and so the cool thing now is take that same idea and apply it to Chitwan Valley Family Study. One of the things I like to think about when in trying to do this is um, just to give you like a metaphor, so to speak, is this is Lake Powell. I don't know how many people know Lake Powell. This is out in the western United States between Arizona and Utah. Um, and for the past 30, 40 years, it has been under a severe drought. And so it's literally hundreds of feet, like 330, 350 feet below its maximum and where they would like it to be a lot of times because of the drought. The interesting thing is like you can see here that white space. You can see a little boat down here at the bottom. It's pretty much just straight down. So it's not a big change in the topography of the lake by just having it go down. But you can still reveal kind of where something is. That is not true when you look at this is Lake Mead, so it's not too far away from Lake Powell. This is near Las Vegas, though. Um, on the left is in 2000 when there was a lot more river water in Lake Mead. And then you see the topography change, right? Where boats can go, where there are islands, where there are peninsulas, where there are rivers, and then it changes over time. The reason why I'm bringing this up is if you think about an environment as kind of like being the water and the underlying rock structure as being the genome didn't change, but the environment reveals certain things, how much of something there is, how little some little water there is. The topography can look dramatically different, right? So a massive change in an environment that affects everyone will reveal different propensities for different outcomes. So Chitwan, and one of the things that was exciting writing this grant that collected the genetic data was a lot of things have changed fairly dramatically. Some of them were political, some of them were social, some cultural. All of those things are changing essentially the water level. And some people are, have the same genetic underlying predisposition to, say, type 2 diabetes or fertility or higher education. But suddenly the social climate has changed, changed for everyone potentially, but some people might benefit from that. Some people might suffer from that because of their genetic predispositions, if that makes sense. So keep that in mind as we think through like how this might apply. I'm not going to go into gene environment interactions a lot because it occurred to me we could have a whole nother lecture just on this. There's a lot in here. So come back. There's several different reviews that have been written on this, and I'm sure that there are several more, but at some point, like, you could find your own things more recently. But one of the things I did want to talk through is just briefly to point out that when we think about measuring outcomes, from negative to positive or environments from maybe harsh to nurturing, um, almost all the gene environment work, and this is still true today, is in the neutral to harsh environment. So how bad did someone have something? How harsh was it? How bad was it for them? And maybe a disease state. So almost all the work is kind of focused in one quadrant. Um, And as you learn more about gene environment interactions, you'll learn there are several different models of testing this. The diathesis stress model is the most common one, and it's because it really only focuses on negative environments or harsh environments and negative outcomes. But there are several other ones to think about related to nurturing environments. And again, I won't get into the specifics of all of the different models, but like a genetic differential sensitivity or vantage sensitivity require positive environments. Um, and even the outcome, very little is looking at positive outcomes, which we have plenty of in CVFS. So there's a lot of room to study that other places haven't really looked at. Um, and finally, kind of talking about um, gene environment interactions, there's actually been a lot more work on testing, how to do formal work for testing interactions. Um, and the thing is, though, that's true for any interaction. Like we get sometimes overhyped on gene environment interactions, but the reality is anytime you have an interaction, you should be formally testing some of these things to be able to distinguish different models. Some other citations if you're interested. One of them being this regions of significance. I would recommend you always do a regions of significance test, whether or not you're doing G by E or environment by environment interactions. And that's just showing where in your data um, there is a significant actual test of interaction because, I mean, as you all know, if you have two non-parallel lines, they will eventually cross at some point, even if it doesn't make any sense. So like, yeah, you always want to test where your differences are. So just keep that in mind. Again, we could do a whole other thing on this, on G by E. The final thing to think about with genetic predictors, and again, you could do a whole other lecture on this or come to our course. We talk about all these things. Is really focusing on that maybe that more biological understanding of genetics and get into Mendelian randomization. This is like Mendel, as in Mendel with the smooth peas and rough peas and things like that. If you remember from intro biology, um, and essentially, this is the same as an instrumental variable, which most social scientists are very aware of. So you might have something that you are trying to derive, like a causal effect between smoking, in this case, your X, and lung cancer. You're trying to prove that, but you can't because you're worried about unobserved confounders. So when you're worried about that, you might try to find something that affects, so a Z that affects your Y only through an X, right? And in this case, that happens to be a tobacco tax, changes smoking levels, and then you can compare, um, maybe people who live in one tax versus a different tax and their lung cancer. Do they have similar or lower lung cancer rates? So instrumental variable, not nearly enough time to talk about instrumental variables. The same thing can be used with your genotype. And there's been a lot of work in this. And actually, Erin, if she's still on, has done some papers on this as well. And so there's actually examples of this with like alcohol intake. There are certain genotypes that change whether or not you can metabolize alcohol. And then that might affect your phenotype. So you could use it as an instrumental variable, or you could start to use the family models as we were talking about before. Ultimately, though, it's always this issue. If you've ever done any instrumental variable work, you always have to be worried about the exclusion restriction assumption. Is it possible that you have some other unmeasured confounder or something that's influencing, that your genotype is influencing other than your exposure? And that's definitely true with genetics. It's complicated, but it's not uncommon for a gene that's affecting, say, alcohol to also be affecting, say, your eye color. And could that be a problem in your model? So a lot to think about here. But what I can tell you is there's a lot of work going on. And they found some pretty straightforward ways for people to be able to apply these in their data without like needing to know a lot of the underlying biology. So and their whole course is on this too. So I put some some examples, some techniques that are mentioned here. Okay, so I'm right on time for what I was hoping for, which is let's talk a little bit about the Chitwan Valley Family Study and the genetic data. And I'm hoping that you all are willing to kind of jump in at some point and talk through some of these with me. Maybe dream up like what you might want to try to do. So this is the Chitwan Valley Family Study CD data. The reason why I'll explain why I'm tying to that, but this comes from the Scott et al. Paper in 2020 that just documents how large the sample is, where it's from, how it compares to the other data, how many people we have in different categories. You can see it's a pretty good mix of age groups. Ethnicity is a pretty good mix, and education levels. The reason why I picked that one is that we collected genetic data from CVFS using saliva as part of the CD study. So 96% of the CD study have genetic data. So that again ties back our genetic samples, and that's why this sample, like knowing the demographics of this sample, is quite helpful. Yeah, and I think that's it for that. So this is the relatedness data. So we have trios, which are really quite unique. You can do a lot of different work on that. That kind of gets you at a causal description of what's going on in genetic data, if there is a causal association, or using it for Mendelian randomization or other types. Because you have a father and a mother, you kind of can adjust out some of the genetic confounding. One parent and one SIB pair, so that's 1,400. A parent-child duo that isn't part of the trios, and even some unrelated. So the point is there's actually a lot of really powerful data here. And if you were to throw out all of the pairs, you'd still have thousands of unrelated individuals that you could conduct analyses on. So there's a lot of really interesting things, a lot of cool things to test. So let's go through some of those. So right now, there is a group as part of that original grant I was talking about, Jordan Smoller and Bill and others, who are creating polygenic scores for MDD, so depression, and AUD, so that's alcohol use. And those polygenic scores, so they're working on those. And we can talk about those as examples of how they might be used to predict, depression or alcohol use. Or we can talk about other polygenic scores that could be created, say related to cardiovascular disease or BMI, educational attainment, Alzheimer's disease, menarche, fertility, all these other things. So this is where I'm hoping that people will start to jump in and either ask questions or otherwise I'm going to start going through how might we test some of these. So actually, I'm going to do something, Heather, that we not normally do. I'm going to save the rest of this slide because this is the last slide. Let me see if there's any clarifying questions first. And then I'm hoping that someone will jump in and say, I want to talk through what the fertility polygenic score might look like. Any questions, clarifying questions? No. Okay. So let's go.
Sorry, can I ask a question?
Please.
Sorry, I'm novice in this, so I just getting introduced to this area. And I'm an entrepreneurship researcher. So
Okay.
So I want to ask, how does the polygenic risk score method differentiate from next generation sequencing? What are the pros and cons to each if I'm considering each?
Yeah, so that's actually in some ways that's a data collection difference. So most polygenic scores, you could create a polygenic score using sequence data. That's no problem. You can do that. The thing is that usually most studies don't have the money to do sequencing yet, although that's becoming more possible. So just so everyone knows, um, sequencing, when we're talking about that, a lot of times what we're saying there is you're measuring every single variant, right? There's different ways of doing it. There's more complicated ways, next generation ways. There's several different methods, but the idea is that you're reading every um, single nucleotide polymorphism. Again, that's that first lecture. We talk a little bit about that. That's the smallest measure of genetic variance that we get to. So we go through each one of those single nucleotide polymorphisms or SNPs. That's sequencing. You can actually get the order of that for every single for the person and on different chromosomes, and it's great. You can do a lot of work with it. What most studies that have done GWASs haven't done sequencing. What they've done is they've used what's called like a chip array, and what that's doing is it has a sample, not a random sample, but a sample of those SNPs across the entire genome. And then it is taking those, measuring those. So maybe instead of every single one, it's measuring one here, and then it's skipping 10 base pairs and going another one, and then skipping 20 base pairs and going to another one. And then it's actually using sequence data to impute fairly accurately, to be honest, like very accurately. Now, the missing data in between those. So it can become closer to, but not the same as sequence data because it's imputing a lot of missing data. So this doesn't really tie to polygenic scores, but in terms of data collection side of things, it's very cheap to do arrays. You can get a lot of data. However, if you have a population that maybe is very, very new, that we don't have good imputation panels for, or maybe if you're really concerned about a rare genetic variant, a rare genetic measure that might not be in an imputation panel, then sequencing is what will always be better because it's essentially the ground truth of what your genome says. If that makes sense, but a lot more expensive because it takes a lot more labor even now to be able to do that. But yeah, these same polygenic scores, you could create these on sequence data. There's no problem with that. Might be, yeah. No, I don't see any reason why you couldn't. Any other?
Yeah, I have one question. Yeah, so what is the sample size requirement to like construct polygenic scores? So I have heard some talk about thousands, like hundreds of thousands, and some also talking about millions. So
Yes,
yeah.
that's a great question. And the short version is if you were doing an exploratory analysis, a GWAS, then yeah, you want hundreds of thousands of people, right? Because you're testing millions of variants. So if you were to, for example, if we were to do a GWAS in Chitwan, imagine that we have 25 or 20 to 25 million variables that we're going to try to test in our sample of 10,000. And let's just pretend that they weren't even related. That's way too many tests for the number of people we have, right? There's no way. We would have multiple testing problems like crazy. But that's for the discovery portion. Polygenic scores don't need nearly that size, right? So now what you're doing is you're taking an external study, you're creating a score. That's one measure, that's one variable. So then it's really only an issue of how big of an effect that single variable has. So if it's something like height, honestly, you could probably construct the polygenic score for height and find a significant hit in 20 people, right? If you're going to do something bigger than that, like, or not bigger, but has a smaller effect size, then like anything else, you'll have to have a bigger sample size. So the short version is for the most part, you can find and we actually did some power analyses on these at some point. I could pull those. I could try to pull those in another talk. You have with many polygenic scores, if there is an effect at all, you will be able to find it in CVFS, unless it's a very rare like disease that you're looking at. For kind of common things, common diseases, common behaviors, this is a very large sample size to find things with polygenic scores.
Yeah, thanks. I wanted to talk about educational attainment and like how we can use, but I think we can do it later.
Yeah. Or we could use it as our example, but yeah.
Yes, sir. Yeah.
Yeah.
In the context of CVFS sample, yeah.
Yeah. No, that's something I've been interested in doing for a while. I saw another chat that came up. Is that? Yeah. Okay. So yeah, if there aren't any other kind of clarifying questions, let's talk through one. Let's pick one. And someone can throw out an idea, or maybe you don't want to because you're like, hey, I don't want to give my ideas to the whole world. So I get that too if you don't want to do that. But let's try one. Maybe not pick one that someone's super interested in. Well, let's try tracing it through the life course processes. Here's one that I didn't list up here, but I think I said a couple different times. One, we could create a polygenic score. I don't think this would be a good idea for CVFS, but let me just test this out and you guys tell me. For type 2 diabetes, right? So let's imagine we take a score for type 2 diabetes, and you could say, well, we have data on now 10,000 people. You saw that we had a couple thousand people who are kind of young adults, and we could say, we know enough about their lives. We know that maybe even what happened when they were born or what neighborhoods they were in, things like that. You could start imagining tracing this through the things that they have experienced, different health issues they might have had. Education is often a good one because we actually social sciences collect a lot of data on that, but there's, for type 2 diabetes, maybe it's something related to exercise or diet or something like that, right? Different changes that they might have. Maybe when they become an adult, do they change their diet? Do they not change their diet? Is it different from other people in different environments? Or in terms of genetic nurture, in this case, what an example would be with like type 2 diabetes is you might say, well, someone's, as a child, you have some predisposition to type 2 diabetes, and you got that through your parents. So that's the direct effect it's the indirect effect from the parents to the kids to type 2 diabetes. But maybe there's something about that score that also changes the way the parent creates an environment, right? Creates what type of food they make, what type of food they select, what type of exercise they do. Again, I'm not saying this is actually a great example for CVFS, but I'm having the realization that maybe we don't want to talk through some of the other ideas that are things that people might want to actually work on. But I'm happy to do that too. So that's an example of tracing it through the life course process. And just think about, for whatever you're interested in, education, fertility, the different measures that we have in CVFS, and how far back in time we have, because a lot of the data we have is longitudinal, so you can see how people have changed over time. So if we did do something like educational attainment, you could, if you had it from someone who was born in Chitwan, you could then see like when did they go to school, when did they stop going to school, what type of school did they go to, and what kind of job did they end up getting. All of those things might be tied to their educational attainment polygenic score even though that was created in adults, many older adults in like mostly European ancestry populations. Selection to different environments, interaction with different environments. Again, same thing. Imagine again that this idea of like a lake, right? If there's a sudden shift in the way, like, again, I'm going to stick with type 2 diabetes because it's the one thing that I don't know that anyone's going to work on here. If there is a sudden change in an environment where sugary foods are more available, or food in general is more available, or maybe restriction of work, or just being able to get outside, it's highly limited, so you're not able to get out as much. How does that change, say, someone's likelihood of developing a disease? So that's that environment where the underlying genetic thing, predisposition is always the same, but the water level is changing and it's revealing different potentialities. Or is it related to, say, again, people who are more likely to go into certain places? One example of this with educational attainment, that from New Zealand at least, was that they found that educational attainment, higher scores of educational attainment were more likely to migrate out of New Zealand. And not just for educational purposes, but for work too. So even if they didn't go to school, they still migrated out. And then finally, there's some really unique models that can be run because of the family tests here. Sibling models or family models that can test whether or not there is a causal effect with some of these. So for example, BMI, is there an actual effect of the polygenic score of BMI on some outcome, BMI or type 2 diabetes, if I'm going to stick with that? Any of those, you could actually test more of a causal model. Now, one thing I'll warn you about is that anytime you start getting into causal models, family models, things like that, as you know, with an instrumental variable, the effect that you would see is kind of in the regular regression is always going to be much larger than the causal effect, right? So if you see a very small effect in like your regular regression, then it might not be worth it to try to test a causal model, because if it's very small to begin with, you're not going to find anything. You know that. If you have something that's a little bit larger, maybe it will. And so some of these things, just as a reminder, going back to the very first lecture, like, again, these were developed in very different populations. So there's a first order interest in just understanding if any of these polygenic scores predict the disease, the outcome, or any of the preceding diseases or outcomes or processes in this population. Just seeing if they predict at all is helpful, is interesting. Seeing if they predict into different environments, or if they're correlated with different environments. Does it predict the same in different, the Terai, Tibeto-Burmese, or some other group? So you could imagine a few different really critical tests that could be done that seem pretty straightforward, but no one else can do them, and no one else has done them. And that's kind of my point, is in some ways, we often make this overly complicated, and we think, oh, I need to see if it predicts this disease and then interact it with these 5 things and then do a causal analysis. And I'm saying, yeah, that's all great. But if you just started off with a simple, here's the polygenic score, does it predict anything related to the disease that it's supposed to predict? Or what is it correlated with? No one else has done that. Novel. It's novel right there. Maybe not the most exciting all the time, but certainly would teach us a lot. of how far this polygenic score is really applicable. Okay, we are at time. I am done. Any other questions, examples? Don't make me talk about type 2 diabetes anymore. But I'm happy to talk about anything else, or please contact me and we can talk through like your own specific example.
I do have a quick question, Coulter,
Yeah,
and
Heather,
it is from your diabetes example, though. Sorry.
no,
But
it's okay.
I
It's
was
just I
just,
didn't know much about it, but yeah, go ahead.
it got me thinking whether or not these scores could ever be used on an individual basis in a clinical setting to
Great
help
question.
people.
Great question. So we talk a little bit about that in the first one, but let me be clear. Right now, the answer to that is no, not any of these scores. Some are starting to work on it to get a little bit better, but and Aaron, please jump in on this if you have any more knowledge. But like people want it to be. They desperately want them to be used in medical situations and clinical situations. One of the things that has stopped, thankfully, has slowed that is that they are not predictive in all populations equally, right? So again, like type 2 diabetes, let's pretend that it predicts incredibly well in European ancestry. It's a even near perfect prediction. If that were the case, then people would want to use it. The problem is that it's not that great of, it's not like a perfect predictor by any means. Um, but also, when you apply it to other populations, it gets cut by anywhere from a third to 90% of that effect. And that's part of what we're trying to do is say we need to improve these scores. When people create new scores, how well does it predict in all these different populations? So I know that one of the things Jordan and his colleagues Bill are doing with the MDD and the AUD are saying here are, scores that we've worked on extensively in other populations. How well does it work in Nepal? And some of them may predict okay, some of them may not. I know this isn't going to surprise you. AUD is very different by gender in Nepal. So it might predict in one, like it might predict in men, but it might not predict at all in women. Why? Because there's actually no variance. There's very little variance in AUD. Almost no women have AUD in Nepal. It doesn't mean that they don't have a underlying genetic prediction. It's just the environment is very different there, if that makes sense. So that's where the gene environments get involved. But yeah, Heather, that's a great question. People really want them to get to a clinical level desperately. Precision medicine is what it's called. If you've ever heard people talk about precision medicine, that's part of this. But we are not there yet, and certainly not in, newer populations to this. Like I would say, Nepal fits into this. But I think that's what we do. That's where we're building the science toward, I think.
Yeah, I was
So,
just gonna add that Chitwan's it's a unique population, and I think that's something that Jordan's been working on quite a bit is adapting the methodology that's used to create the polygenic scores specifically to the Chitwan population, so that they do become more relevant. But I second Coulter is they are not individual numbers at this point.
Not yet. That's a good question, though. And great questions, everyone. We're at time, I think. Heather, do you want to wrap us up?
Yep, thank you so much, Coulter. This webinar will be up on the CVF website, so feel free to share the link.
Yeah.
Thank you, Coulter.
Yeah, thanks. Bye, everyone.